Okay, so let's dive right into one of the most amazing processes in all of life. We're talking about a biological saga that's happening inside you trillions of times a day. This is the cell cycle, a cell's ultimate mission to multiply.
I mean, you've got to wonder about this, right? You get a little paper cut, you forget about it, and a few days later, poof, it's just gone. The skin is totally back to normal. How on earth does that happen?
Well, it's not actually magic, but honestly, it's pretty close. What's really going on is this incredibly precise, high-stakes mission that your body is running all the time, 24/7, to help you grow, to heal, and just to keep you going. So before we get into the nitty-gritty of how it all works, let's just quickly talk about the why. What's the whole point of this cellular mission anyway?
It really just boils down to three main jobs. First, for simple growth. It's how we all got from being a single cell to, well, us. Second, to repair tissue. That's our paper cut right there. And for some organisms, this is the whole shebang. It's how they reproduce.
Now, you can imagine, a mission this important, it needs some serious prep work. A cell can't just decide to split in two on a whim. It has to go through this whole logistics phase first, and it's called interphase.
So if you look at this cell here, it seems kind of quiet, right? Doesn't it? Almost like it's just chilling out, taking a break. But oh man, do not let that fool you. Inside, it's basically mission control at full throttle, running a million checks and getting everything ready.
Think of it like this. The prep work has three stages. First, in G1, the cell is bulking up, loading up on energy and supplies. Then comes the S phase. S is for synthesis, and this is the big one. This is where the cell copies its entire DNA, the master blueprints for everything. And finally, G2 is like the final systems check, where it gets even more energy and proofreads all those new DNA copies.
And this is where things get really serious, because once the cell enters that S phase and starts copying its DNA, there's no going back. It's the point of no return. The cell is now 100% committed to dividing.
But what if that final systems check in G2 finds a major problem? What if the DNA photocopier messed up and the new blueprints are full of typos and errors that just can't be fixed? You can't run a mission with faulty instructions. Thankfully, the cell has a built-in safety measure for this. It's called apoptosis. If the DNA damage is just too bad, the cell basically hits the self-destruct button and aborts the mission. This is absolutely critical because it prevents flawed, potentially dangerous cells from multiplying, which is how you can get some really serious diseases. But let's assume all systems are go. The blueprints are perfect. Now the cell moves on to the main event, the action sequence where it actually splits. It's time for the great divide, mitosis.
Okay, here we go. Prep time is over and the action kicks off with the very first stage, prophase. This is where you can start to see things visibly change as the cell packs up its most valuable cargo.
You can see it right here, all that messy, spaghetti-like DNA. It's called chromatin. It starts to coil up on itself, getting super tight until it forms these little X-shaped things we call chromosomes. It's kind of like rolling up giant blueprints into neat little scrolls so you can move them safely. At the same time, the nucleus, the office holding the blueprints, starts to dissolve. Next up is maybe the most famous stage in the whole process. You've probably seen this one in a textbook before. It's a moment of perfect, beautiful order called metaphase. Just look at that. The precision is incredible. All those chromosomes, each one holding two identical copies of the blueprints, they all line up, single file, right down the middle of the cell, the equator. This step is absolutely crucial for making sure each new cell gets a perfect set of instructions.
And from that moment of perfect, calm alignment, we get to the most dramatic part of the whole story, the separation. This is anaphase.
This side-by-side view just shows it perfectly. On the left, you've got that neat line in metaphase, and then bam! On the right, in anaphase, these tiny little fibers grab onto the chromosomes and yank the copies apart, pulling them to opposite sides of the cell. The blueprints are officially being delivered. Okay, we are in the home stretch now. The final stage of this amazing division process is called telophase. And here we have it, the grand finale. The cell starts to pinch in the middle, kind of like a water balloon being squeezed, and on each end, a new nucleus forms to protect those precious blueprints. The chromosomes can finally relax and unspool. The mission is pretty much complete. And get this, here's the real kicker. This whole incredibly complicated, four-stage dance we just watched, from packing up the DNA to yanking it apart and building two new cells, how long do you think it takes? In a cell like ours, it's all done in about 2 hours.
Isn't that just wild?
So, after all that prep and all that action, what do we have? Let's take a look at the result of a mission accomplished.
And here's the payoff: 2 brand new, healthy daughter cells. Each one is a perfect clone of the one that came before it, with a full set of blueprints, totally ready to do its job, or maybe even start its own mission to multiply. And that's how your skin heals, that's how we grow.
So, as you can see, the cell cycle is just a masterpiece of biological control and precision. But all that incredible precision, it leaves us with one last, really important question. What happens when this carefully controlled mission goes wrong? What are the consequences when a cell just ignores all the stop signs and starts dividing completely out of control?